Okay, so I think we can get started. Welcome everybody and Happy New Year. Thank you for joining us as we start another series of the online Cell Size and Growth Seminar series. Today we're lucky to have two talks, both addressing questions related to nuclear morphology and nuclear scaling. And we're going to start with Simon from Renata Barstow's lab at the Institut Curie. So without any further ado, Simon, would you like to share your slides and get us started?
Okay. Hello everyone. I'm Simon, and today I'm going to talk about whole genome duplication and nuclear architecture. So we all know the famous textbook cell cycle, but in this presentation, I would like to discuss with you the fact that there are actually non-canonical cell cycles that exist and that have been described in many physiological and pathological contexts. But before to talk about these non-canonical cell cycles, I would like to introduce briefly the classical cell cycle, which is based on two main events: genome duplication during S phase and then cell division during mitosis. And these two events are coupled together in order to maintain the diploid status over multiple cell cycles, meaning the fact that cells have two copies of each chromosome, as you can see on this karyotype. But as I said, there are also non-canonical cell cycles. They can be based on aborted mitosis. So this is when cells exit mitosis after or before chromosome segregation, like in cytokinesis failure or mitotic slippage respectively. Or they can be based on the absence of mitosis, like for endoreplication, which is only G and S phases. But they have in common the fact that there is an uncoupling between genome duplication and cell division. So what does it mean? It means that cells duplicate their genome, but they do not divide. And so this is generating tetraploid cells that have 4 copies of each chromosome, as you can see here on this karyotype. And if multiple non-canonical cell cycles occurred, it's generating polyploid cells that accumulate multiple copies of the genome. And so polyploid cells have been described in different contexts, including physiological context. In this case, it's called program polyploidy because cells become polyploid through a reproducible and controlled program. And this kind of polyploid cells have been found in many organisms, including mammals, insects, and plants. And among these different examples are really like the giant neuron of Aplysia over here that become polyploid to become huge in order to innervate a large portion of the body. And when I say huge, it's really huge because it can accumulate 200,000 copies of the genome. And it's actually so huge that you can see one single polyploid neuron over here on a coin without any microscope because the cell size is about 1 millimeter in diameter. But it's a quite exotic example. So closer to you, your megakaryocytes and your hepatocytes are polyploid. And if you are a fly, your salivary glands are polyploid. Polyploid cells have been found also in pathological context. And in this case, it's called unscheduled polyploidy because polyploidy occurs in cells that should not become polyploid. And it has been found in many human tumors, and it's actually one of the most common molecular abnormalities in human cancers, as you can see. But what I found interesting here is the fact that different non-canonical cell cycles can be used in different contexts. And to give you just a few examples in physiological conditions, hepatocytes are generated by cytokinesis failure, while megakaryocytes are generated by mitotic slippage. So what does it mean? It means that different contexts are associated with different non-canonical cell cycles. And so we decided to ask this question: are different paths to whole genome duplication having different outcomes? And because it's a quite big question, we decided to focus first on genetic stability. And so to address this question, what we did is to use human diploid cell lines in which we develop genetic and chemical approaches in order to induce cytokinesis failure, endoreplication, or mitotic slippage. And as a readout of genetic instability, among others, we analyzed DNA damage in the first cell cycle following whole genome duplication. And to do so, we use gamma H2AX here in red, which is a well-known DNA damage marker. And as you can appreciate on these images over here, we observe that whatever the way to induce whole genome duplication, it was always associated with high levels of DNA damage. And before to show you the quantification, I would like just to take a moment to explain you this kind of graph because I'm going to show you a lot of graphs like this during my presentation. So you have three populations. The first one is diploid cells without any treatment. And the two other ones are related to the fact that when we induce whole genome duplication, it's not 100% efficient. So we always have, we still have diploid cells in this treated population that we use as an internal control. And we have also the tetraploid cells, that is the population of interest. And as you can see on this different quantification, we observe similar number of gamma H2AX foci in interface cells, whatever the way to induce whole genome duplication. In terms of mechanism, we found that newly born tetraploid cells actually do not scale up protein amount with DNA content, meaning that they have twice more DNA to replicate, but they do not produce twice more DNA replication factors, generating replication stress and genetic instability. And I like to illustrate this mechanism by this picture of my cat, in which you can see it tries to fit in this small box, but obviously it can't because it's too fat. And it's a bit what happens to newly born tetraploid cells that have too much DNA to fit in the DNA replication machine. But this is a published story, and the main conclusion was that whole genome duplication generates genetic instability independently of the type of non-canonical cell cycle used. But when we were working on this project, we also realized that different non-canonical cell cycles can have different effects on nuclear architecture. And so we decided to focus on this second aspect over here. And we first asked this question: how nuclear architecture responds to changes in DNA content? And to do so, we use the same strategies than before. So human diploid cell lines in which we induce whole genome duplication. But this time, what we focus on was nuclear architecture in the first G1 following whole genome duplication. Okay, so here you have the images. After cytokinesis failure, we obtain binucleated cells as expected, while upon endoreplication, we obtain mononucleated cells. But when we compare the nuclear architecture in these two contexts with the diploid cells, we observe that actually the nuclei are very similar to the diploid cells. However, and in striking contrast, when we induce mitotic slippage, we observe huge nuclear deformations, as you can see here. And again, because I like to use picture of my cat to illustrate my phenotypes, upon cytokinesis failure and endoreplication, it's a bit when it's sleeping like that, it's almost a perfect circle, while upon mitotic slippage, it's more chaotic like that. But of course, I could not use pictures of my cat to quantify this phenotype, so I decided to use another strategy based on these two parameters over here called nuclear circularity and solidity. And to make it simple, when these two parameters are close to zero, it means that you have more nuclear deformations. And so we quantify these two parameters after cytokinesis failure, endoreplication, and mitotic slippage. And as you can see on this quantification, we found that only mitotic slippage generates a significant decrease in these two nuclear parameters, as you can see here. And to have an idea of what happens in terms of cell population, we decided to plot the nuclear circularity with nuclear solidity. And so upon cytokinesis failure and endoreplication, it looked like this. So the two populations of cells, diploid and tetraploid cells, are very close to one because there is no nuclear deformation, and they are very similar, meaning diploid and tetraploid cells have the same nuclear architecture. However, upon mitotic slippage, I think you can appreciate the difference over here. We observe that many cells can have different nuclear circularity and solidity indexes because they have different nuclear shapes. So it means that mitotic slippage generates an heterogeneous population of cells with different nuclear shape, a phenotype that we called nuclear instability. And so the conclusion here was that only mitotic slippage generate nuclear instability after whole genome duplication. And so the next obvious question was, of course, okay, but why only mitotic slippage-induced tetraploid cells have this kind of abnormal nuclear shape? And to answer this question, we decided to focus on mitosis. And of course, there are many events that occur during mitosis, but one of them is the phosphorylation on histone 10. So the histone 3 serine 10 phosphorylation. So this phosphorylation is quite high at the beginning of mitosis and decreased during mitotic progression. And when we were thinking about non-canonical cell cycles, we realized a very interesting thing. So first, when we induce endoreplication, cells do not enter mitosis. So they have always low levels of serine 10 phosphorylation. when we induce cytokinesis failure, cells exit mitosis during telophase, and at this stage, there is no more phosphorylation as well. However, when we induce mitotic slippage, it means that cells exit mitosis with high levels of serine 10 phosphorylation, as you can see here. And so we were wondering if this could contribute to a defect in nuclear envelope formation. And so first, we decided to look if this phosphorylation is maintained in the next G1. So as expected, in diploid G1 cells, there is no more serine 10 phosphorylation in G1, while when we analyzed tetraploid cells generated by mitotic slippage, we were able to find this kind of patch of serine 10 phosphorylations that are maintained in G1, and you have the quantification over here. In contrast, when we induced cytokinesis failure, we did not observe any accumulation of serine 10 phosphorylation in G1. So to be sure that this phosphorylation is contributing to the nuclear deformations, what we did then was to express a mutant that cannot be phosphorylated, so here S10A, and we found that this was sufficient to actually improve the nuclear shape, as you can see here, and to decrease the nuclear instability upon mitotic slippage if you compare with the Y-type condition over here. Then, to be sure that this phosphorylation is at the origin of the nuclear deformations, what we decided to do next was to prevent serine 10 dephosphorylation at the end of mitosis in diploid cells. And when we did that, we observed that the phosphorylation is maintained in diploid G1 cells. So here you have only diploid cells. And more importantly, we were able to reproduce nuclear deformations that are very similar to what we observe in cells after mitotic slippage. And so we concluded that actually upon mitotic slippage, the high levels of serine 10 phosphorylation at mitotic exit contribute to nuclear instability. Okay, so we have nuclear deformation associated with serine 10 phosphorylation, and the next question was, okay, but are these high levels of serine 10 phosphorylation associated with defect in nuclear morphogenesis? And before to show you the result, I would like to introduce just a bit more the nuclear architecture. So you have here the nuclear envelope, which is composed of two lipid bilayers, supported by the lamina network, which is very important for nuclear organization and stiffness. Of course, inside the nucleus, you have the chromatin and histone that can be modified by either methylation or acetylation. And we decided to focus first on histone modification because it has been shown previously that this phosphorylation on serine 10 can affect histone modification levels. So we first analyzed histone methylation upon mitotic slippage, and we did not observe any global changes. However, when we analyzed histone acetylation, we were very surprised to observe that upon mitotic slippage, there is an increase in histone acetylation levels, as you can see here on this different example. And more importantly, we found that this increase in histone acetylation levels is actually dependent on serine 10 phosphorylation, because when we express this mutant that cannot be phosphorylated, there is no more increase in the acetylation level. And more importantly, when we induce whole genome duplication by cytokinesis failure or endoreplication, we did not observe any increase in acetylation level, showing that it's very specific to mitotic slippage and to the serine 10 phosphorylation. And because this kind of histone acetylation was associated with open chromatid and softer nucleus, we were wondering if this increase in tetraploid cells could be sufficient to actually impair nuclear stiffness. And so we collaborated with Sarah Wistrom to perform atomic force microscopy and to measure nuclear stiffness. And we found that upon mitotic slippage, tetraploid cells have softer nuclei, as you can see here. But the next question was, okay, but is it linked to the acetylation levels? And is it sufficient to restore nuclear stiffness to prevent nuclear deformations? So to address the two questions, what we did is to use different strategies to decrease the acetylation levels. And we observed that this was sufficient to prevent nuclear deformations upon mitotic slippage, as you can see here on these two examples. And here you have the quantification showing that just by decreasing the acetylation level, we can prevent nuclear deformations in tetraploid cells, and also prevent the nuclear instability, which is much lower compared to the control condition. So we concluded about this part that the increased histone acetylation levels upon mitotic slippage contribute to decrease the nuclear stiffness and to generate the nuclear instability afterwards. Okay, so we have nuclear deformations due to softer nuclei. The question was then, okay, but how are nuclear deformations generated upon mitotic slippage? And so we analyzed different components of the cytoskeleton because it was known that it was shown that the cytoskeleton can contribute to nuclear architecture. And we found that microtubules accumulate at the nuclear deformations, as you can see here on these images or here on the quantification, suggesting that microtubules are probably involved in the nuclear deformations upon mitotic slippage. But to be sure about that, what we did then was to perform live imaging and to follow cells that exit mitotic slippage over here. And you can see that microtubules are accumulating at the nuclear envelope at mitotic exit, and more microtubules are accumulating here, and more the nuclear invagination is going deeper and deeper into the nucleus, as you can see here, suggesting that the microtubules are actively generating this invagination. But to be sure that it's true, we reproduce the same experiment, but this time we remove microtubules using microtubules depolymerizing drugs over here below. And as you can see on this example, this was sufficient to greatly improve the nuclear shape upon mitotic slippage. And so we found that actually microtubules are contributing to the nuclear deformations upon mitotic slippage in tetraploid cells. Okay, so since now we know that microtubules are generating the nuclear deformations, the next question was, what are the functional consequences of these nuclear deformations? And to answer this question, what we did is a mini screen to analyze different nuclear envelope component or chromatin component, and we compare area in which the nucleus is not deformed or deformed in tetraploid cells. And with this screen, we identified two guys over here, lamina C, which is a component of the lamina network over here, and Escherichia dimethylated, which is known to interact with the lamina network. And so in diploid cells, we observe the expected behavior for these two guys. So lamina C is at the nuclear envelope, and if you look here, we have more, we have an enrichment of Escherichia dimethylated close to the lamina C. This is the same in intact area in tetraploid cells, as you can see here, but we observe a striking difference in deformed area in which we observe much less lamina C and Escherichia dimethylated, as you can see here, compared to the intact area over here. And you have the quantification over here showing that locally at the deformed area, there are less lamina C and Escherichia dimethylated. And again, this is specific to mitotic slippage. When we induce whole genome duplication by cytokinesis failure or endoreplication, we did not observe any of these defects. Okay, the next question was, are these defects resulting from the nuclear deformation or at the origin of the nuclear deformations? And to address this point, we use the same strategies as before to prevent nuclear deformations, either by increasing nuclear compaction or removing microtubules. And we found that this was sufficient to prevent any defect in lamina C and Escherichia dimethylated localization, as you can see here. And you have the quantification showing no more differences over here. So the last question of this part was, okay, but are nuclear deformations impairing 3D genome organization? And to answer this question, we collaborated with Elisa Riccio to perform IC, which is a method allowing to map the interactions inside the nucleus. And here I'm showing you the chromosome interactions. It's tetraploid cells compared to diploid cells, meaning that when it's reddish, it means that you have more interaction in tetraploid cells compared to diploid cells, and when it's bluish, it's the opposite. And basically what we found is that upon mitotic slippage, there are more interactions between long and short chromosomes and less interaction between long and long chromosomes, meaning that mitotic slippage alters nuclear organization in G1 tetraploid cells. Okay, so our working model is here. So we found that upon mitotic slippage, there is more histone acetylation, and this is sufficient to decrease nuclear stiffness in tetraploid cells and to allow the microtubules to induce nuclear deformations and local nuclear reorganization. But what I show you until now was done in unscheduled polyploid cells. And as I said in the introduction, there is also physiological polyploid cells. So the last question of my talk will be, is a similar mechanism taking place in physiological polyploid cells? So to address this question, of course, we had to change our model. We use mouse hepatocytes that become polyploid by cytokinesis failure, Drosophila salivary gland cells that become polyploid by endoreplication, and mouse megakaryocytes that become polyploid by mitotic slippage. And when we analyze hepatocyte and salivary gland cells, we did not observe any defect in terms of nuclear architecture, as you can see here. But when we analyze a megakaryocyte after mitotic slippage, we again observe nuclear deformations. And we thus decided to focus on these nuclear deformations because these nuclear deformations was described, but the molecular mechanism was never identified. And so since the nuclear deformations were somehow similar to what we observe in human diploid cells upon mitotic slippage, we analyzed first microtubules to see if they could be involved in the nuclear deformations. Again, we observed microtubules close to the nuclear deformations in megakaryocytes, and we found that microtubule depolymerization was sufficient to decrease nuclear instability in this context. We also observed more acetylation during megakaryocyte differentiation, and we were very happy to find this paper showing that megakaryocyte nuclei are softer compared to progenitor nuclei. And so we were thinking that maybe it's exactly the same mechanism that explains the nuclear deformations in megakaryocyte. And so we asked this last question: is it possible to prevent nuclear deformations in megakaryocyte by increasing nuclear stiffness? And to do so, we use the same strategies than before. We reduced histone acetylation, and we found that this was sufficient to improve nuclear shape in megakaryocyte and to decrease nuclear instability. Okay, so the take-home message is here. We found that only mitotic slippage is associated with nuclear instability. It means that different paths to whole genome duplication have different consequences in the first cell cycle, and this may influence cell function and so may have been selected to be used in these circumstances. And if we take the example of megakaryocyte, we can think that maybe megakaryocyte need open chromatid to express more RNA and protein, or maybe they need to have a softer nuclei to migrate easily in the bone marrow. And these are questions that we are investigating in the lab right now. Okay, so with this, I would like to thank all the members of my team and my collaborators, and you for your attention. I will be happy to take your question.
All right, fantastic. Thank you for a beautiful talk. And just a reminder to everybody that if you have questions, please post your question or your name in the chat, and then I'll call you out one by one. I'm gonna start though and ask whether you could tell us a little bit about what you either know or think happens when a cell goes through a normal mitosis after a mitotic slippage. Is there any scope for a kind of a healthy mitosis resetting this, or once you have these aberrant nuclei, do they persist even through a subsequent normal cell cycle?
Yeah, it's a very good question. It's something that we tested. So what we did is to induce mitotic slippage, and then we remove the drugs to induce mitotic slippage, and we let the cells perform multiple cell cycles. And actually, we observe that the nuclear deformations are maintained over multiple cell cycles, meaning that even if at some point they are able to perform, let's say, a classical mitosis, the nuclear deformations were maintained. So, but for the moment, what we did is only in fixed conditions. So what we would like to do is to perform live imaging to see if the nuclear deformations are always the same or if it's stochastic in terms of nuclear architecture. But to answer your question, yes, these abnormal nuclear shapes are maintained after multiple cell cycles.
Do you think that result says anything about the mechanisms that drive it, or what is it is there something about the molecular processes that are connected to it that you think that kind of means that it persists?
Yeah, so my guess would be that the cell will use probably, it has been shown at least that you have a given nuclear organization in G2, and this has to be reformed like it was in G2 in the next G1. And since after mitotic slippage, you have nuclear deformations and changes in terms of nuclear organization in G2, maybe the cell will use the same kind of patterns of nuclear organization and will reestablish this kind of abnormal nuclear shape afterwards. But this is something that we have to check. So for the moment, for me, just maybe because cells are deformed. And so since the nuclear architecture has to be maintained over multiple cell cycles, maybe the nuclear deformations are maintained for this reason.
Cool. That's super interesting. Thank you. Next up, we have a question from David.
Thanks so much. Gorgeous talk. Since cancer cells are polyploid and the nucleus is quite deformed, I'm wondering what the kinase is that phosphorylates histone 3 at serine 10.
So it has been shown that it is, so you have different kinases, but the main one is Aurora B. Yeah.
Great. Next up, we have another question from Sarah.
Hi, Simon.
Yeah.
Great talk. So I wonder your hypothesis behind the aging mechanisms, if these can be involved in aging, or do you have any clues on that?
So this, I don't know. Maybe I'm not sure if it has been shown that aging is associated with nuclear deformation. I have to say that
Progeria, for instance, is highly associated. Progeria, that is a premature aging, is highly associated with the deformed nucleus. So I think would be really interesting to look into that.
Yeah,
Thank you.
It could be very interesting, but I think in progeria, what was shown is like laminase mutation, but it could be interesting to look at serine 10 phosphorylation anyway in this kind of pathology. Yeah.
All right, next up, we have a question from Nick.
Hi, Simon. Happy New Year.
Thank you.
I was wondering, is DNA damage or gamma-2HX signal seen in megakaryocytes after mitotic slippage, the way it is in your experimental slippage assays?
Yeah, so we haven't checked in megakaryocytes by ourselves, but what I can say is at least we think that the nuclear deformations are not associated with the DNA damage generation, and we think that for two reasons mainly. The first one is the fact that whatever the way to induce whole genome duplication, it's always associated with DNA damage, even if there are no nuclear deformations. And we were also able to prevent DNA damage, and while we have still nuclear deformations, or we were able also to prevent nuclear deformations, and this was not sufficient to prevent DNA damage. So I don't know in megakaryocytes, but we think that the two phenotypes are not linked together.
And how do you prevent the DNA damage in your experimental setup?
Actually, since we thought that the tetraploid cells do not scale up DNA replication factors with DNA content, what we did is to give more DNA replication factors to the cells to compensate this lack of scaling up, and this was sufficient to prevent DNA damage accumulation in the next S phase.
Excellent. Thank you very much.
So I think next up we have Gabriel, who has, I think, a related question.
You just answered my question, thanks. But I had another question originally, which was also related to histone H3 phosphorylation. Hi, Simon. Very nice talk also. I mean, you said it's Aurora B who's phosphorylating it, but why is it not being dephosphorylated? Like, you have any idea?
I think it's not dephosphorylated because cells exit mitosis prematurely, you see. So it should be dephosphorylated at the end of mitosis, but when cells exit mitosis upon mitotic slippage, it's too early. So I don't know if the phosphatases are not ready, maybe. But what we observe is like, it's not a huge accumulation of serine 10 phosphorylation in G1. So I think it's probably dephosphorylated at some points, but it's not fully dephosphorylated in the next G1. And so we think that this is why we have nuclear deformations afterwards. I don't know if I answered to your question.
Yeah, I mean, I guess you don't know, right? But I mean, the phosphatases are super strong, right? In anaphase, you already see that the pole wire chromosomes are already dephosphorylated, so the phosphatases should be like pretty strong. So is there any remaining active Aurora B even? Like with those microtubules or something? I don't know.
So we haven't checked for Aurora B activity per se, so it's something that we should check. Yeah, it's a good idea.
Great. Next up, we have a question from Dia. Dia, would you like to ask a question?
Hi, Simon. It was a great talk. My question is somewhat similar to what Sarah asked before with aging and, if not aging, going more specific to senescence I don't know if you have looked into senescent megakaryocytes, and if they have a change in architecture because they have reached into that senescent phase. How does that kind of change? Have you ever looked into that?
No, so we haven't checked for senescence in megakaryocyte. So what can I say? We observe that it's not like aging or senescence, but at least what we observe is like more the megakaryocytes are bigger and more you have nuclear deformations, meaning that probably during the life of megakaryocytes, when they are like, let's say, older, you have more nuclear deformations, but we haven't checked for any senescence marker in our population of megakaryocytes, so I don't know.
Okay, thank you.
Okay, next up we have a question from Jinrong.
Yeah, thanks. A quick question that you proposed the microtubule can indent or invaginate and deform the nuclei, but I'm simply curious if the microtubule at that time can also sequester some of HDAX, so you have less level of the isolation in the ground nucleus.
So your question is, could it be due to less HDAC activity? Yeah. So we haven't checked for this. What we try to do is to, of course, to inhibit enzymes that acetylate to decrease the acetylation levels. But we haven't checked for the activity of HDAC or this kind of enzymes. It's something that we should try to explain why we have more acetylation.
Okay, great. And next up, we have a question from Timu.
Great talk. I'm curious about the differences between cytokine failure and mitotic slippage in your data, and what are the factors you think might really separate those so much? And the reason I'm asking this is that I often work with a cell line where we can introduce chemically a cytokinetic failure, but the phenotype looks exactly like your phenotype of mitotic slippage.
You mean you have nuclear deformations?
Yeah, you get these nuclear deformations, you get lack of laminin in the cavities, exactly like you're showing. But in our cells, there is only a single nucleus typically left, so the cells try to separate the DNA, but the nuclear envelope seems to form around all of that. So I'm wondering if you have more insight on, in your models, the differences between cytokinetic failure and mitotic slippage, and which of those differences might cause these phenotypes in mitotic slippage.
Yeah, so the main difference is in our case, I would say it's like, maybe it's too early now. It's like when we induce mitotic slippage, cells exit mitosis before chromosome segregation, so very early in prometaphase. Let me here in prometaphase, so here very before chromosome segregation. But when we induce cytokinesis failure, it's much later on, so it's basically at the end of mitosis. And so I think the main difference, and our model, is the fact that at this stage you have serine phosphorylation, while at this stage there is no more serine phosphorylation. But maybe we can discuss afterwards to compare our methods to induce cytokinesis failure, because it could be interesting for us to test your method, because if at the end you have nuclear deformations and one single nucleus, it could be interesting to look at serine phosphorylation or this kind of things.
Sounds good. Yeah, great talk.
Thank you.
Great. So in the interest of time, I think we're going to have to move on to the next talk now. There's a few more questions, and I will copy them into the channel, the Slack channel that we have for Q&As. So hopefully, Simon, you can go there, answer the few remaining questions. Thank you again for a great talk and a nice discussion, everybody. Next up, it's my pleasure to introduce Evgeny, who's going to talk to us about nuclear to cell volume scaling in mammalian cells. So Evgeny, over to you.
Yeah, thank you, Matthew. Can you hear me well?
Sounds good.
Excellent. Yes. Happy New Year, everyone. It's great to see such a great attendance despite this early post-holiday time. My name is Evgeny, and I'm going to tell you about our project aiming to understand the molecular mechanisms coupling nuclear size to cell volume during cell growth. And for me, it's been a kind of a transition project, because I started this project during my last few months in Jan Schottheim lab at Stanford, and then I continued working on this project in my lab in Cambridge. And I say I, but actually it's Hisashi, a postdoc in my lab, who is currently working on this project. So a lot of the results I'm going to show are produced by Hisashi. As we all know, subcellular components and organelles tend to scale with cell size as cells get bigger and bigger. And even during these seminars here, you've probably heard talks about mitochondria scaling with cell size, ribosomes, mitotic spindle, and other components in the cell scaling with cell size. But still, there is a lot of mechanistic aspects of scaling that are still unknown. For example, how is organelle size regulated is not totally clear. What mechanisms couple organelle size to cell size and organelle growth to cell growth, and whether different organelles in the cell scale differently. For example, we've observed superscaling in lysosomal components with cell size. Does that mean the amount or size of lysosomes will superscale while other components in the cell scale or subscale? There are a lot of questions here, and a good place to start is with the nuclear scaling, because it's a very clear phenomenon. When you look at your cells, the first thing you see is the cell size and the nuclear size. And it's so obvious that even 150 years ago, people measured that and noticed that bigger cells tend to have a bigger nuclei. As you can see on this illustration from 150 years ago, where they looked at red blood cells from birds, frogs, reptiles, and fish, they see this scaling between nuclear size and cell size. And later on, there was another work looking at different fish species, and again, they observed that nuclear size scaled in proportion with cell size. And this was so striking that this phenomenon got its name, karyoplasmic ratio, which is a ratio between nuclear size and cytoplasm size in the cells. And in recent years, there's been more studies there showing that nuclear size or nucleoid, in the case of prokaryotes, scale with cell volume very precisely, and it's a very ubiquitous phenomenon, which makes it interesting to study because it's some fundamental biology that needs to be understood. And in this beautiful work by Paul Nurse's lab, they also showed that within a given species, within the fission yeast species, they can generate cells of different shapes and very different sizes across a wide range of sizes, and they still see a very stable NC ratio across multiple orders of magnitude in size and also across different shapes. So it's a very robust nuclear size to cell size ratio. But the question is, what mechanisms couple nuclear size to cell size? And it's important to answer this question, not just because it's a key aspect of the fundamental biology of scaling, but it also has some practical implications, because the nuclear size and shape can affect gene expression and chromatin structure within the nucleus. And also, for example, we see that the NC ratio is changing depending on the cell type or condition. During cell differentiation, we see that the NC ratio decreases. In cancer cells, we often see increased NC ratio, and that certainly would have some effects on cell physiology. And for example, for cancer cells, it's even been proposed to use this strategy called nuclear size rectification, which means makes the nucleus bigger to prevent cell migration, because if the cell is big and stiff, the cell cannot squeeze through the extracellular matrix or through other tissues, which leads to metastasis. So if we can understand the biology of nuclear size control, we can then manipulate that and achieve some desired phenotypes in the cells. So what we know, what factors set the nuclear size? There has been some studies there in the literature, and one thing that you can think about is the DNA content. Obviously, more polyploid cells have bigger nuclei, but certainly DNA content is not the only factor that sets the nuclear size, because in our body, a lot of cells have the same DNA content, but they still have a different nuclear size and different nuclear to cytoplasm ratio. And also during S phase, our DNA content doubles, but the nuclear size does not double immediately. And we can observe a nuclear growth in the absence of DNA content increase. And I also want to mention this beautiful experiment from 1960s, which is most impressive to me about how nuclear size can change. So in this experiment, they fused a cell with a tiny nucleus, an erythrocyte, with a cell with a relatively large nucleus, a HeLa cell. So they induced a fusion between the two cells, and shortly after that, the bigger nucleus got smaller and the smaller nucleus got bigger, which means that presumably there is some soluble factor in the cytoplasm that limits the nuclear growth. And when the cell gets bigger, it produces more of that factor that leads to the bigger nucleus. There's been also proposed an idea that membrane may be limiting to nuclear growth, and that's also possible. And in addition to that, a few years ago, Joel Lemierre in Fred Chang's lab published their paper proposing that it is actually the pure biophysical phenomenon where the osmotic forces determine the nuclear size in the cell, where the number of micromolecules in the cytoplasm and in the nucleus create pressure on the nuclear membrane and can inflate or deflate the nucleus. So there are many possible models there. And to systematically identify factors that control nuclear to cytoplasmic volume, we decided to perform this high-throughput genome-wide CRISPR screen to systematically identify genes that affect the NC ratio. To do this, we transfected our HeLa cells with a genome-wide library of guide RNAs against different genes in the body, in the cell, and then we sorted cells into the cells with a small nucleus versus large nucleus. After which we can count the sequence and count different guide RNAs to identify genes that affect the NC ratio. But this is not a trivial task to isolate cells with different nuclear to cytoplasm ratios, because we tried to do it with traditional flow cytometry approaches, but we did not succeed, since the changes in nuclear size are relatively small and you can only measure the nuclear linear size. So we didn't succeed at that point, but when we were still thinking about this project, this publication came up where a lab of Lars Steinmetz at EMBL published this technology called high-speed image-enabled cell sorting, where in addition to normal cell sorting, they can image cell on the flight and then sort those cells based on not just on the signal brightness, but also on the shape of that signal or size and different geometrical properties of the fluorescent signal, which was incredible to me because I'm a big fan of flow cytometry, but this one is just crazy that you can combine microscopy and flow cytometry in one experiment. And now it's becoming more and more common, but at that point it sounded like a great revolutionary technology. And in the paper that they published, they for example were able to isolate cells in the different phases of mitosis based on the geometry of the DNA staining, and that's quite impressive. So I packed my pipettes and flew straight to EMBL to work on this project. And this collaboration was funded by the Life Science Alliance, which very kindly supports collaborations between Stanford and EMBL. And yes, in the lab of Lars Steinmetz, and with help with Daniel, who at that point was a postdoc in the lab, we did this experiment where we used a green DNA dye to stain the nucleus and used the area of the nucleus as a metric of nuclear size, and then the area of the cell as a metric of the cell size. Like I said, nuclear size scale with cell size, that's what we see on this plot from the imaging flow cytometry. From this experiment, we gated specifically the cells that either sit below the diagonal and have a low NC ratio, or sit above the diagonal and have a high NC ratio. And even from these images from the flow cytometer, you can probably see that the nucleus occupies a smaller fraction of cell in low NC cells and larger fraction of cell in the high NC sample. And after sorting, I put those cells under the microscope to make sure that the effect is real. And indeed, you can probably see that the nucleus occupies most of the cell here in the high NC sample, which is not the case in the low NC sample. And we quantified that, and we did see that indeed we can separate cells with a very clear difference in their NC ratios. And after that, with the help from Adam, the bioinformatics expert here in the Gurdon Institute, we analyzed the guide RNA sequences in those samples. And just to mention, we had 5 different sublibraries, which means 5 different copies, not copies, but 5 different variants of whole genome libraries. So basically, it's 5 independent genome-wide libraries that we analyzed in this experiment, which gives us a good 5-fold biological replicate. And from these experiments, very encouraging result was even this principal component analysis, where the first principal component very nicely correlated with the NC ratio isolated from the experiment. So indeed, it is a meaningful experiment because we see that we separate cells in accordance with their NC ratio. And from the bioinformatics analysis, we've been able to identify a few dozens of genes that were enriched in low NC and high NC cells. And for those high confidence hits, it was very clear that in each of the 5 sublibraries, there was this clear trend. For example, here, like in all 5 sublibraries, this guide was enriched in high NC cells, but depleted in low NC cells. And here it's the opposite. So we are pretty confident that this result is real. And we started looking into what kind of genes are involved in one category or another. And the first class of genes that we noticed enriched in low NC population of cells were genes related to transcription. You can see a lot of complexes that regulate RNA transcription. For example, a lot of the top hits belong to this mediator kinase module. We see MYC and MAX transcription factors there. And on top of that, we see a lot of mRNA processing and mRNA stability proteins in the top of the list, and also if we go down the list, we can see a lot of those as well. And this is in a great agreement with this paper published by Helena in Paul Nurse's lab, where they also looked at factors that affect NC ratio in fission yeast, and they found that the most enriched family is the mRNA processing and mRNA metabolism genes. So our results are consistent with that, but in human cells. And besides transcription, we also see a lot of vesicle trafficking, which I'm not completely sure what it means, but it could be that it is consistent with the limiting membrane amount, setting the nuclear size. We see mTOR activity, which also can be involved in cell growth and nuclear growth. And among genes enriched in high NC, surprisingly, we saw at the very top a lot of mitochondrial genes responsible for mitochondrial translation and respiration. We still don't know what it means. It's a mystery. So if you have any ideas, you're welcome to suggest something. But we are not touching too much on the mitochondria yet. But we see a lot of vesicle trafficking proteins, mTOR signaling, and also some chromatin remodeling proteins there, which is also not too surprising given that changing chromatin structure can mechanically affect the nuclear size. So after this screen, Hisashi and undergrad student Owen in the lab performed a validation of individual hits. So they picked about 20 individual genes, knocked them out and measured cell size and nuclear size to confirm the results. And from this quantification, where on x-axis we plotted cell size for each knockout and on y-axis it's a nuclear size, we can already see that some knockouts mostly affect the nuclear size without changing the cell size. For example, XRN2 or this CDE1 protein that regulates mRNA stability, they change the nuclear size mostly, but don't change the cell size. While mTOR activity, for example, or RNA-pol3 subunit, they change cell size without changing the nuclear size too much. And we see some vesicle-related proteins as well, which we don't really know what it means yet. And generally speaking, we see that the factors affecting mRNA abundance and stability tend to be correlated with nuclear size change. If they increase or decrease, they change the nuclear size in the same direction. And the translation-related things, mTOR and also some other factors, they tend to regulate cell size, which makes sense. And that is, I think, in a good agreement with the model proposed by Fred Chang's lab that I mentioned in the beginning, where the nuclear size is set by the osmotic colloid pressure between nucleus and cytoplasm. And in the simplest scenario where there is no nuclear tension, which is, I think, the case for the fission yeast that they looked at, the NC ratio is simply proportional to the number of macromolecules in the nucleus, a ratio of those in the nucleus and in the cell. And if we think about it in the aspect in the light of our findings, we can interpret it in the way that nucleus and cytoplasm have slightly different composition of macromolecules. And it's actually in the agreement with this experiment from the same paper from Fred Chang's lab, because mostly those macromolecules are protein and RNA molecules. And when they stain the cells with FITC, which binds both protein and RNA, they see quite a uniform staining in the cell, in the nucleus and in the cytoplasm. But when they treated cells with RNAs to destroy all the RNA, they saw that it was more depleted in the nucleus than in the cytoplasm, which means that in the nucleus, we have a higher RNA to protein ratio than, so it is higher in the nucleus than in the cytoplasm. And we also did this experiment where we stained cellular protein with CFSE dye and RNA with RNA select dye, and we saw the same result, that the protein is pretty much everywhere, and RNA is strongly enriched in the nucleus. This means that transcription, RNA stability, and modification will mostly affect the nuclear size through changing the RNA content in the nucleus, while mTOR activity and translation can affect cell size and NC ratio by affecting the amount of protein in the cytoplasm. So the global balance between transcription, translation, and degradation can set the NC ratio, and that could explain why different cells have different NC ratios. Because different cells have different rates of translation, transcription, and different protein stabilities. And if we imagine a cell type which has a high transcription rate, but relatively low translation rate or high degradation, then those cells would have a larger NC ratio. And vice versa, if you have less transcription but more translation, you would have a bigger cytoplasm and smaller nucleus. And that is well reflected in our screen, where factors that can, for example, promote transcription, they are responsible for nuclear size enlargement, while factors that promote translation, they are responsible for cytoplasm enlargement. And just to make a note, this model, like I said, is in the absence of nuclear tension, but in our case, we also see factors that affect the nuclear tension, such as lamin and also cytoskeleton components. They also affect the NC ratio on top of the osmotic model. You can see this up to 3 complex, which is responsible for actin polymerization, is also very high on our list of nuclear size controllers. And if we believe that it is the osmotic pressure and the ratio between molecules in the nucleus and in the cytoplasm, then presumably we can change the NC ratio by simply shuttling a certain highly expressed protein between nucleus and cytoplasm. And to do this, Hisashi introduced into the cells this fluorescent protein, which can be translocated from the nucleus to the cytoplasm upon exposure to blue light. So you can switch the localization when you shine light on your cells. This is this experiment where we label the DNA with a DNA dye, and here in the red color is this fluorescent protein that can translocate. And when Hisashi shines light on those cells, the protein gets translocated into the cytoplasm, which results actually, as expected, in a decrease in the nuclear area. So the NC ratio decreases when you export that protein into the cytoplasm, which supports this osmotic pressure model. So to summarize, first of all, I have shown you that image-enabled cell sorting combined with CRISPR technology is a very powerful tool for investigating different determinants of cell morphology, and probably you can use it for many things beyond that as well. Second, I've shown that global balance between RNA and protein synthesis and modification affects the NC ratio in human cells. And this colloid osmotic pressure plus envelope, nuclear envelope tension sets the NC ratio in HeLa cells. And we can manipulate that by either knocking out some genes that control the macromolecule ratio or by changing the nuclear envelope tension. And last of all, we have a lot of unknown pathways there, such as mitochondrial translation and respiration, vesicle trafficking, and so on, that don't make perfect sense yet, but it's exciting to look at those things that don't make sense in the beginning. Yeah, with that, I would like to thank everyone who was involved in this work, Hisashi in the first place, but also everyone in the lab, Wendy, Rui, Owen, Ken, and Gunhee. Also, of course, Jan, who really initiated and inspired this whole work, Lars and Daniel, who helped me with CRISPR screen, and Adam, who helped me with bioinformatics, as well as Life Science Alliance and MRC for the funding. Thank you.
Fantastic. Thank you, Evgeny. As before, please post any questions you have in the chat. I'll get us started, and I wanted to ask you a little bit more about what role you think nuclear tension can play in mammalian cells. Do you think it should be altering the scaling properties? Does it mean that scaling might not be perfectly size correlated when you have tension in the system? Do you have some idea about what the model predicts should happen?
I think it can be the case because some literature indicates that larger cells are more active at actin polymerization, for example. And from these results, it looks like the actin works to squeeze the nucleus. When we knock out actin polymerization proteins, we get a bigger nucleus. So I can imagine that scenario where bigger cells would have a different cytoskeleton to maintain the biomechanics of the bigger cell, and that would also change the nuclear mechanics.
Cool. So next up, we have a question from David.
Thank you very much. So in conclusion, it seems like cancer cells are small with large nuclei. So would you say then that cancer cells are transcribing more than they're translating?
Yeah, that's an interesting question. Like, what's wrong with those cancer cells? Yeah, my previous, like how I intuitively thought about it is they just don't have much time to produce protein. They just tend to focus on producing DNA and dividing. So yeah, I can imagine that they just don't translate too much cytoplasmic proteins. But yeah, obviously we don't have any data to prove that, but that's something that can be tested experimentally.
So, I mean, I wonder if you could give an RNase to a cancer cell and shut it down more effectively than if you gave cyclohexamide.
Well, cyclohexamide is tricky. We tried to give cyclohexamide to the cells to change the amount of protein, but it just messes up too many things in the cell, both in the nucleus and in the cytoplasm. But certainly we can measure transcription rate and translation rate in cancer cells versus normal cells and actually see if there is this shift more towards transcription rather than translation.
Great. Next up, we have a question from Wenxiao. So Wenxiao, would you like to unmute and ask a question?
Hi, thanks a lot, Agni. So it's a brilliant talk. I'm wondering, during your CRISPR screen experiment, like how many days you culture the cell between the transfection to the image-based sorting? Because, I mean, if the period is too short, maybe the changes in the NC ratio we observed might be a transit effect. They can recover later, maybe. And if it's too long, maybe it's due to like cell growth or starvation. For example, you have a lot of mTOR or mTOR related genes, like targets you screened, which kind of, maybe related to the cell growth. So how could you balance this thing, like to design the experiment?
Yeah, absolutely. You're absolutely right. The timing is crucial here because if it's too short, the cells don't have enough time to develop the phenotype, completely deplete the protein and change their morphology. If it's too long, you can lose some essential genes there. We kept our cells for 7 days before the actual sorting, and it seemed to work fine. Yeah, we still see a lot of essential genes in the library, although they were more depleted compared to non-essential genes. So the cells were gradually getting those. But because it's equally affecting the big nucleus and small nucleus cells, we still can compare those two from the same population. But yeah, the short answer is it's 7 days and it worked fine.
Okay, so during these 7 days, you arrest them or you just allow them to replicate? To divide?
We just let them grow for 7 days.
So without any drugs, just the wild type medium.
So how the experiment was done, basically the cells have DOX-inducible Cas9 there, and we transfect them with a lentivirus containing genome-wide guide RNA library. So we transfected them, added DOX, and kept them transfected and under DOX for 7 days, expressing Cas9 and guides.
Okay, thanks a lot.
And next up, we have a question from Mimi.
Hi, Evgeny.
Mimi.
Beautiful talk. I was sort of just wanted to piggyback a little bit on kind of the cancer question, but now going to more developmental biology. Basically, do we know enough about like how different cell types transcribe and translate, and see if they essentially, they also follow the same scaling laws, or if somehow, there's subtleties or differences in these different cell types.
Well, I think the short answer, we don't know. Yeah, I think you knew that answer.
But I wonder, I mean, I don't know, you've dug, more than I have, so
yeah. I don't think anyone systematically compared transcription and translation rates across different lineages of cells. But it would be interesting to see, at least during the differentiation time course, right? To see how that changes.
Yeah. Thank you.
And next up, we have a question from Gabriel.
Hi, Evgeny. Very nice talk. So I was just wondering, like, I always thought the scaling with size can be explained by the fact that you're producing more of everything. And when you're increasing translation rates or decreasing translation rates, that you kind of affect everything too, I thought. Or do you think that you have preferential, you have nuclear proteins too, right? It's not just the cytoplasm. So do you think
yes, absolutely.
these in your model, you think you're more specifically affecting cytoplasmic proteins, or
yeah, that's what we think based on this model. Obviously, yes, you do affect both nucleus and cytoplasm, but you affect one more than another. If you change transcription, that would mostly affect the nucleosides. Of course, downstream of that will be the cytoplasm as well, but I think it's about the balance.
You have many more questions. I will not ask more. Thanks.
Next up's Jan.
Hey, the question that I don't know, like looking at the cell fusion experiments where they fuse the cells with different nuclei and then you get the eventual equilibration of the sizes. I was just wondering if we knew what the time scale was of the nuclear size change, and if that time scale would tell us something about the I don't know, like I mean it should be related to these osmotic models, right? So if it's a protein translocation timescale, or if it's transcription timescale, or possibly you could see a two-phasic response where you have the transcription upregulation because now you have in the smaller nucleus and the bigger cytoplasm, etc.
Yeah, I think I need to double check that, but my impression was that the time scale is on a scale of a few hours, which can be comparable to the rate of like protein import and transcription.
Transcription would be faster, right?
Depending on how much you need to transcribe. Yeah.
But again, maybe the transcription is determined by like how quickly you can start importing the RNA polymerase or other transcription factors from the other nucleus. So yeah, so I guess my guess would be somehow dominated by the protein translocation.
Yeah, and slightly related to that, Hisashi now is looking at cells where you manipulate cell size. You know that you overexpress or underexpress cyclin D, and you get bigger cells and smaller cells. In the beginning, when the nucleus just forms around the DNA, right after birth, the size is pretty similar in big and small cells, but within the next hour, the bigger cell becomes bigger. So clearly there is some import dominating in that case. But maybe there is a second phase where transcription kicks in and also hyperinflates that.
Yeah. Really great stuff. Thanks, Evgeny.
Thank you.
And next we have David Lebensky.
Hi. Really fascinating talk. So I was wondering whether it's possible to attach numbers to the nuclear export experiment and whether they make sense. Have you changed the osmotic pressure enough to get the change in nuclear size you see?
Exactly. Yes, that's actually the experiment we're doing like this week. When Hisashi produced this result, I didn't believe it was gonna work, so I was surprised it worked. And now we are really trying to quantify how much of that protein we are expressing.
Okay. Well, I guess I look forward to hearing about that then.
Yeah, certainly. That's something to check.
I think I skipped over Ivan's question, so let's go to Ivan next.
Thank you. Thank you for a nice presentation. My question is, do you see any enrichment of ER proteins in your screen? Because I think that's basically what makes it different from isolated reservoir, which would be kind of like perhaps well explained by osmotic model. But here we always have a surface connected to the nucleus.
Right. Yeah, as a pool of membrane that can be supplied from ER to the nucleus. So we do see proteins that relate to the vesicle trafficking between Golgi and ER. So those are in the screen. We don't see clearly ER localized proteins, but probably that's not something we would expect. But it's more of a trafficking.
It's pretty good.
Thank you.
And next up, we have a question from Sakriti.
Hi, great talk. My question is a little similar. So it's like a thought experiment. So you see two different types of cells, one with high NC and one with low NC ratios. Do you think that when you say that there is more transcription, do you think that this is just an interim phase so that there will be more transcription and then eventually the translational machinery would catch up and then the cell itself would get bigger? Is the ratio kind of like an interim stage? Is that how it's adapting? And also, when you say that there's more transcription, do you think that there is a preference? So, for example, first there would be lamin or ARP2/3, or do you think there's a preference in the priority of transcription?
So I think different cells will just have different intrinsic rates of transcription and translation. If in the end they produced even the same concentration of protein, you can achieve that in two ways. You can express high levels of mRNA and then have relatively slow translation from those mRNAs, or you can just produce a little bit of mRNA but then have a high rate of translation. And I believe different cell types, because they have different composition, they have slightly different basal rates of transcription and translation, and that sets the balance between the two processes. It's not, obviously, if you transcribe one protein, then you get translation and you get a lot of that protein. But it is more about the average rate typical for this given cell type.
All right, so next up we have a question from Shanquin. Shanquin, would you like to unmute and ask your question?
Sorry, hi. Fantastic talk. I was just wondering whether your light-induced protein translocation experiment has been done in the reverse way, because I suppose that driving more protein from the cytoplasm into the nucleus is a more uphill battle. Really, it gets to the question, how much more protein do you need to put into the nucleus to get that effect? Right? Thank you.
Right. Yeah. We did not do it the opposite way per se, but we did turn off the light in this experiment and let the protein translocate back. And in fact, this reverse process was a lot slower than the nucleus. Like the translocation happened fast, but the nucleus size failed to change as fast. So there must be some additional adaptations, for example, flow of membrane or something that adapt in the cells. But we do know from the work of Fred Chang and Gabriel here that when they overexpress nuclear localized protein without a translocation, just an NLS or NES, they can change the NC ratio by expressing high levels of one or another.
Thank you.
And next we have a question from David.
So in the hen HeLa experiment that you titillated us with at the beginning of the talk, is the hen erythrocyte nucleus transcribing more at a faster rate than the HeLa nucleus? Is that why the two equilibrate?
I don't think anyone measured that. I mostly thought about like nuclear import of proteins into that nucleus. But it could well be that, but it's harder to imagine how you can induce such a huge burst of transcription. I was more thinking about the nuclear import.
Thank you.
All right, and so I think the person who asked the next question has logged out. So I'll post that question to the Slack, and we can finish with a question from Curie.
Hi, thanks for the talk. It was really nice. I'm just wondering, I'm really interested in in your idea about the possibly different ratios of RNA to protein in either the nucleus or the cytoplasm. And I guess ribosomes are, I guess they count for like a large fraction of both RNA and proteins in a cell. So if there's like some kind of global change in RNA protein ratio, do you see, or are you interested in any change in how ribosomes are synthesized or assembled? Because maybe proteins are exported and not enough ribosomal RNA is in the cytoplasm. Thank you.
Yeah, that's a great question. What kind of RNA we care about, right? It's not just the mRNA. The largest amount is probably the ribosomal RNA. And it's hard to tell. We do see, for example, RNA pol 3 in our knockout screen coming up as a hit. And RNA pol 3, it would polymerize tRNA, rRNA. So those certainly have a big input, but whether it's a big input in the nuclear size or is it a bigger input in the cytoplasm size through translation, it seems not clear yet. We are going to look deeper into that, specifically into different kinds of mRNAs, different kind of RNAs, mRNA versus ribosomal RNA. But so far, I cannot give you a clear answer.
Thank you.
Great. Well, thank you again to both of our speakers. I think that concludes all the questions that we have. Somebody asked,
great questions. Thank you.
Somebody asked something about the recording, but we'll send out emails with recordings of the session. So that will be for anybody who wants it. Thank you again to both of the speakers for a great start to the series, and everyone for all of the really nice questions. We look forward to seeing you again in 2 weeks' time for the next iteration. All right. Cheers.
Great.
Thanks, everyone. Bye-bye.